So this is the more fun bit, I guess. So once you have assembled, or at least, sort of created one file for all of your Fastq files and put them in the most appropriately correct order, you can then start to do whatever it was that you planned to do with your data in the first place. So in this, I'm just going to, as I say, show you some options. Obviously, whatever you want to do with it is entirely up to you, but there's some options in terms of programs. Epitome and Epitome Labs are slightly different, but I'll explain that. So you might want to annotate your genome. You might want to know where the genes are, or introns, or chromosomes, if that's the case, and you will need to annotate that. And so you can use a program called Procker, and Procker annotates bacterial genomes fairly quickly. And so that will hopefully provide you, I guess it depends on what your genome is and how well covered it is, but it will attempt to identify genes. And I think it uses a reference to sort of help to annotate that. That could then potentially go on to something like GeneBank and then be utilized by other people. Variant calling is a really useful one. So if you're wanting to look at differences between isolates, so looking at single nucleotides, for instance, this is individually within a text reader, or you can use viewers. So there are different variant calling programs. So Medaka, again, I've used Medaka and that seems to work quite nicely. And you can use human or bacterial calling. Or there's also a program called BCF Tools, which has more options and is a little bit more complicated. I certainly found Medaka was easier to understand than BCF Tools, but you might find that one is more appropriate to what you want to do. And again, there are plenty of others as well. And I have actually put a link to a list of loads of different tools in the PDF. So as I say, you might find that whatever I'm saying doesn't necessarily fit exactly what you want. Have a look in that list and hopefully you'll be able to find one that is more appropriate. Once you've made your variant calling files, you can then view them in genome. So there's IGV, there's Artemis, and Artemis has a comparison tool. And they basically, you can put your sort of start isolate genome in and compare it to the genome that you're looking to identify, and it will provide sort of colored changes where the SNPs are. So they're quite useful. And there are lots of different viewers as well that you can use. Phylogenetic trees are also something that a lot of people want to do, especially so you can either look, for instance, for differences between, say, a ward outbreak of microbe-resistant bacteria, or if you're evolution, there's obviously many different uses for phylogenetic trees. But they generally fall into the programs fall into two different categories. One that uses maximum likelihood and one that uses Bayesian methods. The maximum likelihood ones tend to be easier to use because they have less parameters to work out, and you do have to have a kind of a basic understanding of phylogenetics and evolutionary biology and how these things work to make it work well, I think. But maximum likelihood tends to be easier, and there are programs such as RaxML, IQtree, and Fastq you can use for that. And then Bayesian is Beast, but that requires other programs within it, such as Beauty, which processes the data, and then Tree Annotator and Figtree to sort of view it. So there are different ways of doing phylogenetic trees, basically, many different ways. If you wanted to identify plasmids, so plasmids, if you're looking at antimicrobial resistance genes, plasmids are useful because they are obviously mobile genetic elements that carry resistance genes. It can be quite difficult to separate genomic DNA and plasmid DNA, but there are programs that you can use to do that, and Beat Plasmid and MobSuite are two that you can use. So there are also databases and servers that you can use to analyze your data. And there are pros and cons to using them, interfaces rather than command line interfaces. So a lot of people find them easier to use. That's a big pro, I think. Some of them do utilize Fastq files, and then the database processes stuff for you. Some of them require an assembly. Some of them require Fastq files. They're useful for certain applications. There are quite a few that are based on looking at tuberculosis resistance gene identification, for instance. And they don't necessarily need a high-powered computer to run them, because what you do is you upload them to the internet, and the internet cloud does the processing for you. Although there are sometimes rules about file sizes. In the common categories, some of them do require full assemblies, so you will need to go all the way through the last section and process it to get your assembly. As I mentioned, some of them you can only upload certain sized files to. Some of them have graphical user interface versions. Some of them also have command line interface versions. And if you understand how to use the command line, some of them you can bypass the file size limit, because you can do it on your own computer. I think TB Profiler is an example of that. But yes, you just have to be aware that some of them can only do a certain amount. And they can only analyze what they have been designed to analyze. So as I say, TB Profiler, for instance, only analyzes tuberculosis resistance genes, and it can only identify genes that have already been put into the database. So if you're looking for a very new gene, resistance gene, for instance, I don't know, maybe linezolid, it won't necessarily be on there. Or if you're looking for something that might not be on there, so you have to be aware that there is a limited range of analysis with these. And you also have to be careful with what you upload. Obviously, if you are taking a sample from, or if you're taking DNA from directly from a sample, even if you try and remove the human DNA from that sample in the laboratory, you may have human DNA within that sample still. And obviously that's a bit of an ethical issue if you're uploading human DNA to the cloud. You can bioinformatically remove human DNA as well, but obviously the idea of these databases is that you don't have to worry about doing all of the sort of line commandy kind of stuff. So just be aware that what you're uploading can be limited by the ethics of what you're doing. Here's some of the database exhaustive, let's say, what is one for instance. There's Pangolin, which is for stars, lineages, variants, finder for resistances, TB Profiler, and micro, both TB ones. And then there's Nadaro. I've put a list of useful databases in the PDF. But what I do want to show you is Epitome. So, as I mentioned, Epitome Labs and Epitome are slightly different. Epitome Labs is basically a cloud-based system that shows you how to go through processing data. Epitome is a program, or at least a cloud-based program, that kind of does it for you. So it's a database and it has certain workflows. So if you wanted to do certain things, then it's quite useful for that. So for instance, it has an AMR one where it looks for AMR genes, WIMP, which means what's in my pot. So if, again, you had a sample, say a sputum sample, and you didn't know what species was in it, you could use the WIMP workflow, 16S. There's loads of different ones, and I can show you in a second. So you download a desktop agent, which is where you then put your Fastq files. So you can just put your raw Fastq files into Epitome, and that's what's really useful about it. You don't have to worry about going through processing it all. And then there's an online dashboard where you can view and download the reports and more information about it. So let me show you that. So this is the desktop agent. So I've got this as a button on my home screen. You need to sign in. You need to have an account with Nanopore, but you can press new analysis. And where do I go? Let's see, I've got that. So if I go to folders, I could put in my Fastq file and go to next. And then it gives me the option for all of the different analyses. So I have starred, because I do this a lot, Fastq, AMR, and what's in my pot. But if you go to all, you can see that there's loads of different things that you can do. 16S, human stuff. Yeah, so you basically click on it. You have to tell it, are you uploading human DNA? So if you have, as I say, a sputum sample, for instance, it won't want you to put human DNA in because of the, unless you have ethical approval and stuff. So ideally it's better to do it from an isolate, and then it will tell you where you want to put the data and information, and then you can press accept and start, and that will run an analysis. And it can take a while because obviously it depends on the speed of your internet and how much people are doing at the time. And it will give you, so this is the AMR information, so it will tell you how many reads you've got, how many alignments, and information about the different genes. But you can also look at this on the Epitome Dashboard, which is online. So you just type in epitome.nanopore.com and it will bring up this page here. So you've got your dashboard. So these are all of the runs that I've run recently. And if I just click on open report, it brings up information. So I think this is from a different project. So this isn't TB. I think this was from an AMR gram positive and negative one. But what it will do when it's finished loading is it will show you the different resistance genes that this has. It will also give you basic information about the number of reads, the total bases that you've got, your quality score, how well your sequencing went, so how many reads you got over time. Obviously with nanopores, the pores die off, you'll get less and less. But it gives you lots of useful information about how many of them pass the quality score and the sequence length. So it's kind of gives you the QC information as well, so you can bypass that. With WIMP, what's in my pot? So this was an isolate, so it's saying that the majority of my reads look to be Citrobacter, but obviously if you've just put Fastq files in, especially if they're really short sections, and as I say, this one is native, so I might have a couple of reads that might have been only a few hundred base pairs, it might then decide that some of them might not be Citrobacter and it might think they belong to something else. So if, as I say, I've got 33,000 reads that it believes belong to Citrobacter, so I can be fairly confident that this is a Citrobacter. But as I say, there are some, and it will be closely related species, and you can change the cutoff as well in terms of abundance. So as I say, the higher it is, the more specific it is. Obviously, if you have lots of species, it will create an actual tree. And as I say, if you had multiple Fastqs that you wanted to create a phylogenetic tree for, you could add them all in and create an actual phylogenetic tree fairly quickly and easily. The AR genes found, so you can change. For instance, you can tell it, I want only clinically relevant ones, and you can change various things about it. So this epitome is a really useful way of looking at data quite quickly and quite easily. You can print the reports, you can send it to people. So yeah, I quite like Epitome. It's very useful. What else have we got? Oh yes, and also if you go to workflows within Epitome, it just explains about the different types of workflow. If we give it 5 seconds, if I can set for WIMP, for instance. Clearly my internet
whilst it's loading. Oh, don't
so
worry,
if I
it's
wanted
loaded.
to, yeah, it's done it. It's because my internet's probably quite slow. So you can open an example report of what you should see, or you can look quick view or learn more, and it will explain more about what it is and how it works. So as I say, it will explain what's in my pot is a workflow, blah, blah, blah. And so it will just help you identify whether it's the right thing for you. So yeah, Epitome is very, very useful, especially if you're not particularly confident with line commands. So yep, there's that. And that's it. We've raced through that. But hopefully what everybody has learned today is now you have a basic understanding of the bioinformatics and the commands that you might need to analyze Oxford Nanopore data, how to process Fast5 and then Fastq files and how to QC them, how to then assemble those files into one big assembly, and then what your potential options are for downstream analysis. And as I say, how to use Epitome, which is a nice, easy way of doing that. And that's something that Nanopore have created with Metricore, I think, for this exact reason, for people who don't necessarily have a lot of bioinformatics skills. So there, I think we've done. Does anybody have any questions about any of those things?
You have the mixed up, yeah.
Okay.
Can I ask something about kind of data ownership?
Sure.
And when you use like Epitome, do they say if they are like, do they own the data with you now, or is it still yours?
I think, I'm not exactly sure. I think Vicky told me this once, or maybe it was Alf, that once it goes onto the cloud, I mean, it still belongs to you, but I think because you have uploaded it, it then stays there. So this is why they ask you, is there human DNA? And you tick the box. So I'm not exactly sure, and I can look it up, but I think it might be that any cloud-based thing, it's not sort of, it's now on the cloud.
Yeah.
But I will look that up for you.
Thanks.
Thank you for a very interesting talk, Lindsay. A question regarding the QC average. What's the cutoff of that? So I see in the example that you showed us, it's 10. What would be acceptable? What would not be considered as acceptable?
So the reason I think that failed is because Oxford Nanopore generally sets the Q score at 7, and I think Illumina sets it at 10, and that's why, because it seems to be set to Illumina, it fails, because it's not that it has failed, it's passed the QC for Nanopore. So the quality score is kind of arbitrary, because when you get to starting your run, you can change it. Generally, it is set at 7, and so that, I think, is the kind of general quality score that people sort of abide by. But I guess it depends on what it is that you wanted to do. I suppose perhaps if you were really specifically looking at SNPs, you could make it greater, and therefore it would be more accurate. Or if you were purely just looking at what have I got, is it an E. coli, then you could maybe lower the score and you would get more past data more quickly, and therefore you could more quickly identify what species it is.
So is it dependent on the species that I'm looking for, or the kit I'm using?
As I say, you can set the quality score when you set Minnow up. As I say, it's generally set at 7. That seems to be the arbitrary sort of level that is acceptable in terms of quality. But I guess it depends what it is that you want to do with your data, or what sort of the downstream side of things is. As I say, if you were specifically looking for really small individual changes, you might want to up the quality score to ensure that your data is more accurate. But if you were just looking purely, as I say, for species, and that's all you wanted to know, as soon as it comes across a section of sequence that is definitely E. coli, it will flag up as E. coli if you put it into Epitome and WIMP. So you can run Epitome at the same time as running Minnow, so you can pretty much have real-time analysis of certainly of what's in my pot kind of things.
Okay.
Does that answer your question, sort of?
Yeah.
I can definitely look it up more though.
Thank you.
I have another question if no one else does.
Sure.
Have you had experience with like concatenating Fastq files? Because you know sometimes Illumina
Yes.
it will give you like read 1, 2 or whatever.
Yeah.
Yeah. What's that like? Is it difficult?
So I used, and Pactome will be able to tell you because he and I did it together. I forget what the code was, but it's a fairly simple, just Ubuntu based, or it might even be Windows. I can't remember off the top of my head. I don't know where I would have put it. So I have million. So this is where I keep all of my command line documents. And I think I should have somewhere. So here we go. Just moving it down so I can actually see it. So demultiplexing, there's one. Trimming barcodes, there's one. Combining Fastq files. So this is the command that you can use to do that. So if you move into the folder where all your Fastq files are, and then press type, and then star.fastq, that means do this to everything. And then your output, you can call it whatever you like. Obviously, I decided to call it barcode1 combination. That will output a file that has all of your Fastq reads in it. So instead of, I don't know, say 4 Fastq files with 4,000 reads, you will have all of those 4 times 4,000 in one file. It's not in the right order. That's the difference between assembly and just concatenating. So you will just have randomly placed thousands of reads, but then you can put that into your assembly programs, for instance. I think that's, so that says Windows. So however, so I think it might be an Ubuntu command or a Linux command. It might work on both. I can't remember now off the top of my head. I think I used Windows.
I actually managed to do it myself with concatenating, but it felt so easy. I was like, this must be wrong. But I think
yeah,
that's fine, actually. Yeah.
So yeah, I mean, I agree. I thought I would have to use some actual program, but you should just be able to do it because all it's doing is telling the computer to just copy and paste everything into one file. So it's in theory not actually too difficult in terms of commanding. Yeah.
Perfect.
Does that make sense?
Yeah, very good.
Great.
I have a bash script which I can provide you for Illumina to do this.
Yes, Sylvia.
Oh, that's amazing. That's a great gift.
Because the problem with Illumina is that you have 96 samples. Each of them may be in 2 or depending on the platform you are using, up to 8 files per sample. So it's automated, and at the end it names the file with the same sample name as you have put in the sequencing file name.
That's perfect. That's what I have, Sylvia. I have 8 files for each sample.
Yes.
And, okay, that's really good. Thank you.
I can provide this, don't worry.
Do you want me to show you how to get onto Epitome Labs, or has everybody kind of managed to do that already? Yes, no? If it's not helpful, if you've done it already, then that's fine.
I've done it already. I'm not sure about everyone else.
Great. Well, I can show you anyway. Why not? Epitome Labs requires you to download Docker, which is a container program. So I'll just start up Docker. So that's Epitome Labs running now. This is because I've obviously already got it set up, and there's quite useful instructions when you go onto the quick start page. You then need to have this Epitome Labs launcher, which sometimes takes a little bit of time to go. So I click it multiple times and then I've got 15 versions of them. So just going to give it a second to load up. As you can see, my entire desktop is now full of various sequencing-based programs, Microbe, IDV, Minnow, RexML. This is my life now. Come on, you can do it. Come on, a little quicker. Should have opened this up earlier, shouldn't I, really?
It's really nice, actually, Lindsay, that you showed us both how to do it with Ubuntu and Windows. It's good. Does that mean you're running both? You've got an Ubuntu laptop now?
Yeah. So
okay.
No, I've got a Windows. This is what the WSL is, the Windows subsystem for Linux. It's really useful for, I mean, it's easier if you have a Windows computer to run Windows, because you can just, as I say, copy and paste file name, file paths in. Whereas, okay, that's why. I've now also got Ubuntu, and that obviously you can only use certain programs with Linux. So Linux and Mac OS are much more similar, and I think Sylvia, please correct me if I'm wrong. Most of them, if they run on Linux, you should be able to run on a Mac, but Windows is more different. And so if you run the Windows subsystem, you can then run a sort of, I suppose it's almost like a separate container that has Linux-based commands in. So I run it using Ubuntu. And it just means that I can run programs that I wouldn't, like, for instance, Fly and Minimap 2. I couldn't run this on a Windows computer if I didn't have the Windows subsystem.
Yep, yep.
So you should get this. And if you press open Epitome Labs, this will bring you into the server. And if you go to, so yeah, this is the start page, and you can see, hopefully, at some point, it will bring up all of these different, there we go, notebooks. And so these notebooks are really useful because you can see that it will explain how to assemble basic QC tutorial. So if I click on that, it actually explains what you're doing and how to do it. It explains the type of programs that you're using. But then also, when you get down to it, it explains the actual commands. And so you can run that command by pressing play, and it does it for you. So it's a really nice middle ground between not knowing commands and then knowing how to actually use the individual programs. So it uses the same programs, as I say, wherever it said it was. It uses Minnow or Guppy to do this. And if I went on to assembly tutorial, it says it uses Fly and it, oh, whoops, it uses Fly and Medaka. So it's really good for, sorry, I can't see it because the bar is in the way. So it's really useful for explaining how to do it, but it shows you the actual commands for the programs that you need to use. So as I say, it will tell you at the top, you're assembling sequencing data using Fastq files, using the Fly assembler, and then you're correcting and polishing it using Medaka. And it will also tell you what you need. So you need to run this Epitome Labs server, so this page, and you will also need at least 16 gigs of RAM, so that's quite a lot. But if you went on to the basic tutorial, you should see you need at least 8 gigs of RAM. So with Guppy, you can do stuff on a normal computer, but if you're starting to assemble, because it's such a powerful program, you need a much more powerful computer. And as I say, there's lots of different things that you can do on here. There's a SARS-CoV analysis, variant calling, various different things. It's very good. It's very useful. I thoroughly recommend, if you haven't done already, to download it. It's a bit like, I think, Galaxy, if anybody has ever used that, but it's specific for Epitome, specific for Oxford Nanopore sequencing. Yeah.